Hello and welcome to Microbiology Shorts. These are short videos on microbiology topics by Rebecca Payne, that's me. Today we're talking about enzyme function and what enzyme function is affected by. There are often other molecules that are bound to enzymes, and these come in two major categories. One category is cofactors and the other category is coenzymes. Cofactors, and both of these are generalizations, but for our purposes, the definitions work. So cofactors are non-protein molecules, most of the time inorganic molecules, such as a mineral, that enable an enzyme to function. And generally the enzyme isn't going to function well or at all without the cofactor bound to the enzyme. Some of the minerals that function as cofactors include zinc, Zn, Cu, copper, Mg, magnesium, Fe, iron. Coenzymes, on the other hand, are organic molecules, and these are generally vitamins, but they can also be other small organic molecules. And they're bound to an enzyme, and oftentimes they sit in the active site. And when they sit in the active site, they complete the active site. So you can think of a coenzyme as a missing piece that makes the active site the active site. It makes it the right shape, it makes it the right size, it makes the substrate able to bind, whereas without the coenzyme, the substrate often isn't able to bind. Vitamins like B1, niacin, and folic acid can function as coenzymes. Of course, there's other coenzymes talked about in your book, things like NADH and FADH2 and CoA, which is short for coenzyme A. Other things that affect enzyme function include pH, temperature, substrate concentration, and something called inhibition. So let me talk just briefly about pH and temperature. So pH, both high and low pH, are able to denature proteins, which unfold the protein. And when you're an enzyme, you're a protein, of course. And when you unfold as an enzyme, you lose your active site shape, and that causes the enzyme not to function very well. So there's an optimal pH for every enzyme. Temperature as well has an optimal range. High temperature is also able to denature proteins. And the thing that's not on this slide, but which is very interesting, is that low temperature does not denature proteins. Low temperature will actually preserve microbes. You can actually freeze bacteria and store them, and when you thaw them out, if you thaw them out properly and give them nutrients and a good temperature, they will actually grow up again. So long-term storage of microbes is done at low temperatures frequently when you're dealing with bacteria. Upper range of temperatures, though, if you denature a protein by putting it in boiling water or subjecting it to steam for long periods of time, denaturing a protein is normally irreversible if there's enough heat applied. If we look at these in terms of graphs, here we have enzymatic activity on the y-axis, we have temperature in degrees Celsius on the x-axis. Sometimes temperature in degrees Celsius is hard to figure out, right? So we're going to say that just a little over 20, about 22, is about room temperature, 70, 72 degrees. 37 would be over here, is about body temperature, human body temperature, 98.6 degrees. Over 50, you're talking really, really hot. Down to 10, really, really cold. So this is an example of how an enzyme would work inside of a bacteria that's subjected to different kinds of temperatures. You notice that really, really cold temperatures, the enzyme activity is virtually nothing, and it steadily increases with increasing temperature until it reaches a maximum where it plateaus. So this is the optimal temperature for this particular enzyme. Somewhere, I'd say between 35 and 40 degrees Celsius is its optimal range. Above 40, it takes a steep, sharp drop down to 50, where it doesn't have any activity at all. At this point, it's going to be fully denatured, deactivated, and not work anymore. pH as well has this kind of a bell curve shape. So enzymatic activity again on the y-axis, pH on the x-axis, and pH goes from 0, which is extraordinarily acidic, all the way to 10, which is extraordinarily basic. About 7 is neutral. This particular enzyme looks like it's functioning from about maybe 3.5 to perhaps 7-ish, somewhere between 6 and 7. And So that's its optimal range, which is actually fairly wide and fairly low for an enzyme inside of a microbe. Microbes generally have optimal pH ranges that are up in the neutral range, so say from 6.5 to 7.5. So this one's fairly low example. Maybe this is an acidophile, which would enjoy acid pH. And the last in this group is enzymatic activity versus substrate concentration. And this is a different graph than what we've seen before. So if you add increasing amounts of substrate and you saturate or flood the enzyme with a whole bunch of substrates, then you're going to get a rapid increase in the enzyme activity, but eventually it's going to plateau. And that's because at this point the enzyme is working at its absolute fastest rate. It can't go any faster. It's like a toll booth worker on the Benicia Bridge on Thanksgiving weekend, when the cars are backed up into Walnut Creek. Those cars, they can't get through any faster because those toll booth workers are working absolutely as fast as is humanly possible. And once they get to the point where they can't work any faster, well, the activity levels off, and the substrates, the cars, they build up behind and tend to pile up. All right, so we'll talk about inhibition in a later short, but thank you for joining me for this microbiology short, and have a great day.